Okay, I will start. I'm Enguang from Zan's lab, and today I want to share our recent modeling of sampling of macromolecular complexes in the minimal cell. So collaborating with JCVI, we started the modeling of minimal cell um, from the two most important subsystems, the first stochastic gene expression and the deterministic metabolism back in 2019. And together with the, oh, let me, uh, together with the 3D structure reviewed in cryo-ET, uh, we simulated the first 20 minutes of the cell cycle in 4D. And now we are able to simulate the whole cell cycle with the full dynamic and also the partitioning of chromosomes over the entire cell cycle as presented by Zan last week. And today I want to focus on another important subsystem, which is the complex formation. So in thing 3A, key micromolecular complexes needed essential cellular functions, along with the central dogma, RNA polymerase in transcription, ribosome in translation, and also deguadosome in degradation of mRNA are all monomeric particles. And important membrane complexes like ATP synthase, ABC transporters, they import the actual cellular metabolic precursors for the cellular activity. However, the investigation of macromolecular complexes assembling remains rare, mainly due to the complicated landscape or the assembling pathways of each complex, and also the different dimensionalities, uh, meaning the 3D cytoplasma and also the 2D membrane, and also the tight coupling between the gene expression and also the complex formation in vivo. So on the right is uh, a classic assembling of a important ribosome small subunit 30S in a parallel network. Starting from the Bayer 16S RNA, 21 ribosomal proteins will bind with to form the final product 30S small subunit. And in our current model, we decided to reduce this whole parallel network to a linear pathway with the largest fluxes. And just another taste of these membrane complexes. ABC transporter for nucleoside called RNS-BSAD import nucleoside from extracellular media that would be converted to the nucleotide. And it is well known that this ABC transporter has a well-defined composition or domain composition of substrate binding protein, transmembrane domain as permease channels, and also nucleotide binding domain as ATPase. Our previous model simply assume a representative species, and in my current model, we are going to explicitly assemble this complete complex. There are a series of bimolecular reactions. Assume a spatial homogeneity with no explicit diffusion and also no modeling of cellular compartments in a 3D space. And we're also using a hybrid stochastic deterministic algorithm. The gene expression and complex formation are simulated by chemical mass equations stochastically, and also and the metabolism simulated by ordinary differential equation. And we communicate these two algorithms per second to synchronize these two subsystems and to simulate the evolution of the entire cell. And so this simple model can be efficiently parameterized to simulate 100 cells within 1 day. This brings advantage of like testing different parameters and also like to validate some assumptions. Um, so before diving into the complex formation, I want to introduce the cell growth and gene expression to set up the context. So our model predicted a cell cycle of 102 minutes, resulting from the transmembrane biogenesis, transmembrane protein biogenesis, and also the lipid synthesis from metabolism. The protein synthesis is intrinsically stochastic. You can see the random birth of mRNA and death of mRNA from translation and degradation. And at the end of the cell cycle, we are able to double the proteome, which is crucial for cell division and also these synthesized proteome or protein subunits will constrain the following or the downstream complex formation. Okay, the first example is the ribosome biogenesis. A ribosome is the largest complex of 20 nanometer in diameter, and each subunit, small and or large subunit, are ribonucleoprotein complexes. So in our simulation, large fraction of cellular resources are devoted to the synthesis of ribosomal subunits, meaning the RNAs, mRNA encoding ribosomal proteins at transcription and also the translation of ribosomal proteins. And at the end of the cell cycle, we're able to completely assemble 400 ribosomes. And you can also see a a considerable fraction of incomplete small subunit and large subunit due to the waiting time for subunit synthesis in gene expression and also the time for biomolecular bindings. And from a simulation, we can also define the bottleneck subunits, meaning the subunit with least counts, then limit the formation of the whole complex from the supply side. So in our simulation, 23 sRNA is the dominant limitation, which is reasonable given the manner of the transcription happens of the RNA operon. So another missing part from our previous model is the translocation network for proteins to membrane. So specific gene targeting sequence will emerge from the ribosome exit tunnel during the translation that will be recognized by either signal recognition particle or Sec-A, and then which will direct the whole complex to the Sec translocon sitting embedded on the membrane in a co-translational translocation manner. And we predicted the targeting sequence of transmembrane protein or lipoproteins using bioinformatic predictions. And after incorporating this whole translocation network, we predict 9% of ribosome residing on the Sec translocon, which is consistent with the cryo-ET observations. We also responding to the very interesting report from James and John last week. We think this could serve as a mechanism for the incorrect trafficking of cytosolic proteins, where 26% of the genome are secreted to the surface, especially those highly expressed like EF2 enolase. And our proposed scenario here is the cytosolic proteins are mistargeted by Sec-A or RSRP, then to the membrane, then glycosylated by unknown transmembrane proteins. One advantage of this scenario is that the cytosolic proteins will be secreted through the narrow channel of the Sec translocon while unfolded. And another support of this hypothesis is that the N-terminus of in enolase in B subtilis is important for secretion reported like years ago. We think Sec-A or SRP selective ribosome profiling could be a very important could be a quantitative experiment to conduct to verify or testify this hypothesis. Okay, so and as you see in the last picture, so the membrane complexes assembling is constrained to the 2D membrane, and the association on the 2D membrane has been proposed a long time ago to benefit from the increased effective concentration due to the reduced search space on the membrane, and also the greater reaction probability due to the constrained conformation of the membrane proteins. However, the absolute 2D rates of the association was seldom quantified until a recent paper for membrane anchored particles. Here we decided to vary these 2D association rates of transmembrane proteins and also peripheral membrane proteins, spanning three orders of magnitude. And from our simulation, you can clearly see that the unassembled accumulation of intermediates will increase dramatically resulting in the decrease in the assembled membrane complexes at the end of cell cycle. We also derived a formula of the unassembled fraction of synthesis monomers of a simple dimerization reaction to show that a relatively fast association on 2D compared to gene expression is required for the efficient assembling. So one last point we want to highlight here is the co-expression of proteins, especially for the most complicated membrane complex ATP synthase, comprising of 21 subunits in 8 unique kinds, encoded by gene from 0789 to 0796. In our current model, we approximate the expression of 8 subunits independently by introducing the proxy promoter strains at transcription reactions. And consequently, you can see the population median of the produced subunits was roughly maintained in stoichiometric balance. However, the non-stoichiometric gene expression at single cell level cannot be avoided. For example, a single cell that short in subunit epsilon will undermine the assembling of ATP synthase in this single cell, and the and at and as a result, the maximum possible assembled ATP synthase was dramatically reduced from 108 to 77 due to this deficit. And we argue that we will introduce the co-expression of operons and considering the RNA isoforms from the long-read and short-read transcriptomic studies that we collaborated with the JCVI. And at last, we want to very briefly share our ideas and plans on extending our whole cell model to multiple cell cycles. So, for example, by extend the simulation to 20 cell cycles, we can simulate 2 physical days. And by doing this, we can investigate the cellular activities upon the depletion of nutrients like glucose and nucleoside. We're also interested in the inheritance of some like metabolic phenotypes and variability across cell generations. And resonating with JCVI, we are also interested in the aging and senescing resulting from the intrinsic factors such as the accumulation of damages and abnormal cell morphology that reviewed by the cryo-ETs, and also the asymmetrical cell division. And we have done a very preliminary attempt to simulate five cell cycles covering 200 cell replicas with assumed constant media. And the simulation didn't explode, which is very nice. And we observed a greater variation of protein counts, cell cycles. And this attempt also reminds us of some missing components of our simulation, like of our model, like the regulatory mechanism in metabolism. And with that, I want to thanks for your attention, and I'm happy to take any questions.
I am just dazzled by the things that you are able to do. So, Enguang, I know that you are like for the ATP synthase, you're basing your thoughts there on transcriptome data, but you've also got now brand new quantitative proteomics data.
Yes.
Does your modeling based on transcriptomics agree with what we observe in terms of the quantitative proteomics, knowing that membrane-associated proteins are the proteomic results are a little less reliable, of course?
Yeah, so we haven't incorporated the transcriptomics data yet. We base our proxy promoter strains on the proteomics counts of the peripheral membrane proteins. That's the answer, yeah. But we do see some distribution of mRNA isoform for this ATP synthase operon from the long read analysis conducted by Troy, yes.
So what's the experiment that we could do for you that you would most like to see right now?
Oh my God, that's a good question. I think We already have the very good analysis of transcriptomics from Troy,
Mhm.
one set of long read and also short read transcriptomics from Paulson's lab. Me personally, I personally think the ribosome profiling will be very helpful considering like it bridges the transcription and also the translation and can do a cross-checking between the transcriptome and also the proteome. And actually, I proposed this idea to Zan, but as we discussed, it's hard to find a collaborator to do this sort of experiments for us. Yeah.
I will talk about it with my team, and we need to meet with your team soon. Any other questions for Enguang? James?
Beautiful work, Enguang. Related to your
Thanks.
point about the non-stoichiometric abundances, I'm curious how you're taking that into account when simulating the assembly formation. And I'm going to
Mhm.
drop a paper into the chat that is a theoretical framework by Arvin Mordegon and colleagues
Mhm.
that describes how the non-stoichiometric ratios can actually help complex formation because it can help
Oh,
you avoid kinetic
okay.
traps.
Yeah, yeah. Is that
And
a
exactly, yeah,
2015 Nature Communication paper? Is that paper 2015 Nature Communication paper? We, yeah, so the
exactly,
short,
exactly.
yeah, yeah, so we consider this situation. Actually, the example proposed by that paper is the ribosomal subunits of large subunit. And we do observe the non-stoichiometric from the proteomics of instinct 3A for the ribosomal proteins. However, for ATP synthase, that's not the case for because there are other studies using like ribosome profiling to show that the expression of different subunits is very proportional to their stoichiometry in this complex.
Exactly. You're
So
thinking of the work by Jean-Wei Li and colleagues?
yes, yes, that's a
Yeah,
very
okay.
good review paper. Yeah,
Cool, cool. Okay.
and so for ribosome proteins, that's also one feature we observed, and we made some assumptions there to avoid the kinetic traps because it's very so basically we consider kinetic traps for only few cases because we don't know what the interaction is inside the cell. So we assume no kinetic traps to for the best affinity or efficiency of complex formation. Yeah.
Brilliant. Thank you very much.